where do we go now? That's the central question of our time. You know, we did the social distancing. It looks like everything's going away, at least temporarily. But the mystery that everybody has, and no one knows the answer to this, you know, is the epidemiologists tell us that if we go outside and start to mix, everything's going to get really bad again. We got to be very careful, which may be true. There's no way saying we just don't know. That's the thing, we just don't know. But on the other hand, you know, what we could say is that if it's attenuating a little bit, maybe a lot, that maybe we can kind of alter kind of what we do and kind of alter our thought process a little bit based on whether it's attenuating. And so, you know, so the prediction is that if you go start looking around, not only will you see mutations that are more virulent, that give more glycosylation, like D16G, D614G, but actually ones that make less, you know, and in fact, that's starting to happen. So what's happened now is that there are spike protein mutations that are being found. We found a preprint from a bunch of guys in Taiwan, I think it was, who had done this. And, you know, we looked at, they posted about 13 mutations they had found. Again, these are hardcore bioinformaticians. I'm not one, but they posted these and on a preprint. And when you look at them really carefully and you say, how do they affect NXST sites, these you know, hypothetical glycosylation sites, these canonical glycosylation sites, how do they affect them? And in fact, in 2 of them, they make it more virulent, one of them being D16G, D14G, or D614G, and the other ones being less glycosylation. And in fact, there's 2 of them that you lost a site. There was 1 or 2 of them that predicted for decreased glycosylation. So, you know, the theory kind of makes sense. It kind of fits a little bit. What was also interesting is that a bunch of guys in China, again, this hasn't been published yet as far as I know, it's in a preprint, they took a bunch of viruses out. You know, they not cultured them, because when you culture the virus, it makes its own mutations. So you really got to be very careful. You know, you got to take the virus as soon as you take it out. Don't put it in culture, but just dump it right onto cells so it doesn't mutate, so you can get an idea of how virulent in culture it is. You know, either looking at the cytopathogenicity in culture, looking at kind of, you know, the viral load that you get, you know, after, like, I think 24 and 48 and 72 hours. You know, so you can kind of see how virulent they are with those definitions. And what happened is that they had a variance. They had, I think, 11 or 12 cultures from these guys that they took out of patients in Wuhan, and they had a variation up to 270-fold in their virulence. And it was interesting. So the ones that looked more like the ones that were in Washington on the West Coast of the United States were very non-virulent at all. I mean, they were virulent, they did stuff, but again, this is cell culture. Who knows what they do in human beings? But on the other hand, they definitely, you know, they grew less. The cells didn't look as bad when they put them on them. There was less viral load, as opposed to the ones that look more like the ones in Europe. You know, that they were closer on the phylogenetic tree to the ones in Europe were, like, you know, in the case of one of them, 19 times more, one was 200 times more. Now, there's a bunch of different mutations of these, not just the spike protein ones, but, you know, it kind of all makes a little bit of sense about what's going on and explains things. So, you know, clearly, it looks like, at least with a first approximation, that there are multiple strains of the virus circulating around, all of which have a slightly different amount of virulence. Now, viral evolutionary theory, you know, and there's a lot of it. It's a really interesting theory that's been going on, suggests that when you introduce a virus into a new host, it attenuates over time, and when it makes a jump to a new species. And the experiment that was done was actually an interesting one. It had to do with these rabbits in Australia. And if you've ever been to Australia, they're very strict about quarantine. You can't bring a lot of stuff in because they're an isolated island that has its own ecosystem. And when you introduce things into their ecosystem, they go crazy. And so the rabbits were introduced probably in the 40s somehow, maybe in World War II, who knows. But they were overrunning, I believe it was Eastern Australia. And they were overrunning Eastern Australia. So biologists, I think in the early 50s, released this virus. It was called MXYV. It's a classic experiment. And initially, all the rabbits died. I mean, it was 100% fatality. And it worked really well. But they found over time, I think it was a period of months or maybe a year, so it was fairly quick. I think it was months, and you have to go back and look at it. But the bottom line, what happened was that the rabbits started living. The next few generations of rabbits started living. And in fact, they would get sick with the virus, but they wouldn't die. They still, a certain percentage would die, but the death rate wasn't as high. And so these guys spent like 50 years or 40 years. It was a classic experiment. It kind of lasts into the 80s. It's now got a revival. You know, they looked at the mutations. They saw that you get a bunch of different attenuating mutations. They all converged to an attenuated phenotype. So it was the phenotype that was important, not the genotype of the virus. And it was interesting. So they've kind of followed this along over the years, and it's actually I guess a 60-year experiment already. They're still publishing on it. You know, the last couple of years ago, there's a guy at Penn State who's actually looking at this very carefully. And it turns out that occasionally the virus pocket, they come out. So in other words, you get like a burst of the virus. The virus figures out what the animal's doing, the animal reacts, and it's like an arms race. One guy does the other. But the bottom line is the most important thing about this is that the viruses seem to attenuate. And that was a really important issue, and it still is a very, very important question with SARS. What really happened to SARS? And I think there are people who say, listen, we did really well with quarantine, and we were able to do case, tracing and isolation, and we got rid of it. But on the other hand, there's another group of people, and it's very controversial, that says, listen, there may have been attenuating mutations in this. And in fact, they found a nucleotide deletion in a protein called ORF8 that's actually, believe it or not, I think at the end of the day, involved in kind of the expulsion of virus and processing of virus into the lysosome. But in other words, they found a deletion in ORF8 that actually, could potentially theoretically predict for attenuation. In fact, they tested it in cell culture a few years ago and found that this is exactly what it did. It blocked viral replication somehow. So again, and it happened very early, though. The key here is it happened very early in the production. It was early in the SARS epidemic. And so that's really important to understand, that it's not only progressive mutation, it's that you've got all of these different ones out there and there's competition. Okay, so it's a founder effect. You've got all these multiple different founders. And what happens, the more aggressive ones burn out because they kill people, right? If you're dead or you're incapacitated in the hospital, you can't spread it. And so that was happening. And I think that's what's happening. And if you look kind of New York City and that environs, what was happening, you look at Italy too, it was the same thing, was that the people who really were getting infected were the healthcare workers. That you had a bad, you had this kind of D614G thing, and you were a healthcare worker, and you had it and you got really sick and you were in a hospital and you were intubated, or even they didn't even know it at the time. And this ER guy thought you had pneumonia and they brought you in and they got infected, the case people and the people working in the emergency room, and so those people seem to be a lot of the only people toward the end of the epidemic who are at least getting infected more than anything. And there's still, obviously New York is still happening to some degree that we're going to have to try to figure out. But again, when you put this all together as a virus that infects the dendritic cell through DC sign and the pneumocyte through L sign, a lot of this makes sense. Now, again, this is all theoretical. This is coming from a medical oncologist, for crying out loud. I'm not a virologist. I'm not an infectious disease guy. I'm not a card-carrying immunologist. But I'm working with a pretty good immunologist who I think, and he and I think, really think this is a reasonable explanation for